Yes. May I restart?
Yes.
Odd things? Okay, sorry by the interruption of the audio. I don't know what is the portion that I stopped to talk, so I will talk all the things again. Okay. First of all, thank you very much for all of you to watch our symposium. This is the last part of our symposium after 2 days, and there are three more lectures of our international internship talks with our students. Uh, but I would like to say first of it and announce our first speaker that we have the certificate of acceptance of the symposium, and please, it is a remind you to not forget to fulfill the assignment form to get the certificate. Okay, it is in the description of the video. And the second thing is I'd like to thank the people that are sharing their experience in the symposium. Rafael Castro, that was one of our speakers in the morning, Paloma Torrati, and the Laboratory of Bioecology of Crustaceans, and the Laboratory of Palynology at Jabuticabal, UNESP. Thank you very much. And I'd like to share you just a photo that is very cute. This is our student Paloma Torrati watching Professor Wilfred Klein in the Jonathan's Code speak this morning, and with her little baby watching our symposium also. It's so cute. I have to share this photo with you. As Professor Thomas said yesterday, the program is a big family, and this is true. Okay, now finally, Natalia, we are going to announce our student, Natalia Rosigari Alves Costa, that will lecture her theme from cosmopolitan to endangered native fauna, comparative biology in the land down under. Natalia, thank you very much for your time and for all the patience with our technical problems.
No worries. Okay, so I'm gonna share my screen with you. Please tell me if you can see it. Okay. Can you see it, Milton?
We are seeing a screen, but not your presentation. Now.
What about now? Okay.
Yes.
Okay,
Perfect
so
now.
can I start?
Please.
So hi everyone, good afternoon. My name is Natalia. I'm a PhD candidate at our graduate program in comparative biology. And today I'm gonna share with you a little bit of my experience as an overseas student. So I did 1 year of my PhD in Australia. And the title of my lecture is From Cosmopolitan to Endangered Native Fauna, Comparative Biology in the Land Down Under. So why the Land Down Under? That's the name Australians call themselves because oftenly Australia and like us, South America, we are always in the down under part of the world map. So they do this kind of joke there, putting the world map upside down. And that's why I brought this picture for you, so we can see a different vision of the world. Okay, so how I ended up in Australia. in Brazil, my advisor is Professor Tiana Kolzer. So I'm a member of the Laboratory of Evolution and Integrative Biology here at USP. And Tiana, while she was doing her PhD, she got to know Professor Robbie Wilson, which nowadays is the PI in the Performance Lab, as a professor at the University of Queensland. And because they knew each other, Tiana invited Robbie to give a lecture a few years ago in 2017 at the seminars that we usually have in the program Café Consciência. And that was an opportunity for me to know him, and we talked a little bit about our interests in science, what we were studying. And then I got the opportunity to apply for the Doutorado Sanduíche no Exterior. So I get a CAPES grant fellowship to go study there for 1 year. And here's a very lovely picture of us celebrating at our beloved and missed old Invicta, which is not the same nowadays. So this was a celebrating time because I already had the fellowship by this time. So before I talk to you about what I did there, I would like to share a theoretical background and the main questions that my research project has here. So basically, I'm interested in climate change and how this affects animals, traits like physiology, behavior, and morphology, which is specifically true for ectotherm animals, which are hardly influenced by climate changes. So my main focus of research is morphology, and specifically locomotor performance, which is important for animals' basic activities in life, like foraging, escaping from predators, reproduction, and survival. So my research I have the aim to answer if lizards' morphology are being affected by temperature changes over time, and if temperature variation during development influences morphology, and if so, what are the consequences to animals' locomotor performance? So this is basically my thesis. My work name is Influence of Environmental Parameters in Lizards' Development, Ecomorphology, and Locomotor Performance. And some of my results you can see in my video, which is here in the Comparative Biology channel at YouTube. So this is what I did so far here in Brazil. When the opportunity to go to Australia came, I had the idea of make a connection between climate change and morphology in Australian endemic lizards. I do the same thing here with a Brazilian lizard, which name is Stropidurus catalanensis, the species. So I traveled and went to study at Wilson Performance Lab with the main question, lizards' morphology are being affected by temperature change over time. So the city that I lived there for 1 year was Brisbane, in the state of Queensland, which is this amazing city. The landscapes are breathtaking. I really love to live there. It's an amazing place to stay. And more specifically, I developed my work at the University of Queensland, which is this also amazing building. This is a picture that I took there when I arrived. So this is my first picture at the university. And I can't say anything about UQ, the name of the university is the University of Queensland, without talking about the purple rain. So when I got there, it was spring. And I was surprised by these beautiful jacarandas trees that they have there everywhere, especially in the university. So it's a very lovely memory that I have from UQ when I see these pictures of the jacaranda season. And my building was really nice too, because I used to work here at the Goddard building. So this is the biological sciences school building. And every day I had the opportunity to look at Darwin and Mendel and say, hi, folks, how are you doing? And get through this door to do my work. So it was quite nice. Coming back to the title of this lecture here, I decided to talk a little bit about my experience with Australian animals. So the first part of the talk will be about the cosmopolitan ones. So to study the lizards that I was interested about, I had to work at the museum. So I worked for a few months at the Queensland Museum. And Robbie got me there, and we have a conversation with Patrick Cooper, who's the curator at the herpetological collection, and also Dr. Andrew Amy, which is a researcher there. And then I told them that I would like to do quite the same work as I was doing here in Brazil. So I needed to work with a species that would be widespread around the Australian territory, but also from natural and urban areas. So the one that was chosen was the Lampropholias delicata skink. It's from the Syncytiid family, which has this very diverse, it occurs in a variety, in a range of places there in Australia, so it was perfect for my purpose. So like I said, I spent a few months at the museum. The lizards were tiny, more tiny than I expected. So the first challenge that I got when I was there was to separate males from females, which is a nice thing to say because here in Brazil, my lizards have sexual dimorphism, but these ones didn't. So I have to open them and try to find some ovaries or tests. So it was quite an experience because I never opened an animal and looked for organs like this before, especially in a different animal collection. And after I did that, I ran some tests and I found that these animals, they don't have any sexual dimorphism. So this was my first result, which I wasn't expecting. And after that, I started taking the 18 linear measurements in 242 specimens. So this is an example of the measurements that I took there. And after I collected all the morphological information, I united this with the climate data. So I entered in this site, the Australian Government Bureau of Meteorology, to see if I could find any data about temperature, mean temperature, minimum temperature, average temperature, and my point I would like to remember, it was that I would like to link differences in the temperature, for example, with the overall body size of these animals. So after I did that, I went back to the university to analyze my data. So I spent another few months working on that. And the results that I brought here for you today, well, this one is about the relationship between mean temperature and snow vent length. So it's the size of the lizard. And I found no relationship. So whether the temperature is high or low, it doesn't mean that the lizard will be shorter or bigger. And then after I took these measurements, I went for the other morphological measurements. I ran some PCA tests. And again, I did the test comparing with the temperature. And I also didn't have the expected results. Like this is the hypothesis that I had, but we are not seeing any difference regarding the temperature and the size of the animals. So Lampropholias delicata morphology has been affected by temperature over time? Not exactly. So now I'm writing my thesis and I'm reanalyzing all this data to see if I can extract any more information that would be interesting because I have a huge amount of data. So I'm at this step now. So this was the first part of my work. I was working on this for about 8 months there. And then when I was about to come back to Brazil, like a couple months last, I had another opportunity with the second part of this talk that I want to say to you about the endangered native fauna. So one day So Professor Wilson, he works with a variety of subjects. So nowadays, he is really focused on soccer players, on the performance of soccer players. But he also studied a lot of animals during his career. So here are some examples of his websites. He worked with optimal performance, science of skill, and also with a really nice topic, which is conservation ecology. So they are working with conservation ecology for about 9 years now. And this was one work there that I did a collaboration with the people from the lab. So one day I was sitting in my desk and Robbie and Sky came and said, Natalia, would you like to go on a field work to a remote island in the Gulf of Carpentaria, where you can only get by a really small and quite dangerous airplane, which is true, because here you can see a man, and you can see how tiny this airplane is. So it's frightening. A place whose beaches are surrounded by crocodiles, saltwater crocodiles, brown snakes, and death adders, which are one of the most venomous snakes in the world. Work with conservation ecology of super cute endangered marsupials, like this guy here. And I was like, yes. So that's how I end up at Groot Island. So I travel all the way here from Brisbane to Darwin, and then from Darwin to the Groot Island to basically work with conservation ecology, help on a ongoing work that what people were doing there with the northern call and the northern brown bandicoots, which are these cute little marsupials, which are endangered there. So they are threatened of extinction. But Groot is also home for a variety of other really nice animals like the dingoes, the monitor lizard, the frilled neck lizard, which is really fine, and this bird that builds this nest and then covers with it with lots of colorful presents to attract female. This is how he looks like. And of course, cute wallabies. So this is the size of the island, so it's quite small. And the first thing that got my attention when I got over there was the fabulous landscapes. The beaches were amazing. They have their own environment there. It was really nice. But something much more interesting is that this island is land of original people, so native indigenous people that still live in Australia and have their own place there. So these are a few women from the community. They do this amazing work with dyeing tissues. It's amazing, it's really nice. And then my work there consisted on a busy day that I'm talking a little bit to you right now. So we were there for capture and recapture of these animals and to look for some ecological information about them. So remember that they are doing this work for 7 years now, so they have lots of information about the island. So the first step that I did when I got there was setting traps. So a trap is this thing here, where I used to put some dog food there to attract the quolls or the bandicoots. And we left the trap there overnight because they are nocturnal animals. And then we oftenly get back there at 6 a.m. to collect any animals that were in the trap. And getting back to the lab, which was actually our rooms, in the place that we were staying, it was the time to do some measurements like overall body size, weight the animals, collect some poop, collect some hair for DNA samples, and sometimes the animals were recaptured. And with this data, they are able to say if the population is growing, if there's movement of they are not reproducing quite well. There's a difference between wet and dry season, so it's a very important work. And I never worked with something like this before, and I got in love with them. I fell in love with this work, which was really nice. So one of the things that we did was look for little babies at the mama pouch. So these are cute little ones that were attached to the mama's teats. And after that, after lunch, I was collaborating with another PhD student at the lab. So her work was regarding performance, qual, bandicoot, the other mammal performance. So to do that, we need to build an arena. So I oftenly spend all my afternoons building this arena where we put the bandicoot and run some performance tests with them. So we recorded the behavior and how he moved overnight. So it was quite a nice work because, again, I never built anything before going there. It was an amazing chance to learn a different kind of English because I didn't knew by the time the name of the tools that I was working with. So this one is a star picket, which was really nice for me. And here are some pictures of me with the bandicoots. So this is a bandicoot and this is a quoll. And this is an example of the experiments we did there. So we were recording the performance of these animals. So here are some cameras recording how fast he runs and everything. And this little cloth here was another experiment that we were doing that. So we attached an accelerometer to these animals and then released them in nature. So we could have access to how long they can walk and how fast they can run. So it was really nice. After I came back to the island, it was almost time to come back to Brazil. I was extracting data that we get there, and this turned into another parallel project between me and Robbie. So by the time it's called individual variation versus between individual variation in northern quoll performance. And so I collected performance data regarding motor control, grasp, bite force, jump, sprint speed, maneuverability. And to do so, I have to learn some new skills, like working with software that I never saw before. And like this one, which is Tracker, which was an amazing experience because when I came back to Brazil, I started my work here with lizards locomotor performance. And then I already had a lot of experience in this software, so I'm quite a tracker pro now. So I thought this work in the island was changing my professional and personal life because it brought to me lots of different information and experiences that I would never believe that I would have someday. So this is a picture of people that work there, the land rangers, and everyone that allowed us to work with that native animals because this is a very remote island in Australia, so it's not a common destination for people that work there or go there on a vacation. So I'm really thankful to Anandiliaqua Land Council and the Anandiliaqua Land and Sea Rangers. And again, I want to remind you that this is Aboriginal land, and every time that I would say anything about Groot, I will say that Island is home of the Anandiliaqua people. So this is a very important lesson for me. And talking about some personal experiences, when I was in the island, I get to do a beach cleaning day, which was really nice because we got to the beach, like a very remote beach, to just get some rubbish that was brought to the island by the sea. So it's not the case of, okay, some people went there and just left some rubbish there. No, we were collecting things that came from the ocean. So people who know me knows that I'm a little bit concerned about pollution and plastic pollution. So I just thought this was an incredible opportunity for me because this is the amount of rubbish that we got there. This is Georgie. She is one of the rangers collecting some of the samples. And also another thing that was really nice that I did there was I went to some caves and I had the opportunity to see these paintings. And I think it's nice to remind you that Aboriginal people were the first people to leave Africa. So they left Africa about 65 or 40,000 years ago. And since then, they are the true owners of Australia. So I think this is an important thing for us to remember. And when I was there, I had the opportunity to do lots of things. And I think one important thing to do when you are overseas or you have the opportunity to work with a different lab or with different people is to like hug every opportunity that you have. Like, so this is me in a conference that we spent 3 days in a conference at an island close to Brisbane. So this was the Biological Sciences Annual Conference at this research station, which was amazing. I learned a lot. So I talked to people that I never talked before. People were studying like clownfishes, lots of other subjects that we are not used to. So I thought it was a nice opportunity to learn from other people. And also, the University of Queensland, they give some free classes of an amount of themes. So I learned a lot about RStudio, like 3 modules that I never had the opportunity to do here. I learned about Illustrator, 3-minute thesis, which was something that I never heard before, and point evidence explanation link, or PEW, which is a writing course. And also they have Friday lectures, which is quite the same as our Café Consciência. So it was nice because we were just there listening to some other research. So it was nice. And to finish my presentation, I would like to remember you that when I stayed there during the year of 2019, and when I left, it was November 2018, and I think you all remember what was going on here in Brazil. So Australia is 20 hours difference from Brazil. So almost every day when I woke up, it was a surprise to see what was going on here. And one of the worst surprises that we got there was the cut in the fundings by the Ministry of Education. So we joined all the other students that went to the streets at the 15M day. And this is the picture of us showing that we were really concerned about what was going on here in Brazil. So I would like to finish my presentation with this picture that it's nice for me because I see my field boots like this one, talking about all the adventures I have to my other shoes. And I would like to say thank you for everyone that was somehow involved in this. Thanks, Tiana, for the opportunity. Thanks, Vera, because Vera helped a lot when I was doing my application. Thanks, everyone from the coordination. And my friends that was there, oh, okay, you should go. And if you have any questions or would like to ask something, maybe here we don't have this opportunity, you can send me an email. And this is what I wanted to share with you. Thank you very much.
Thank you very much, Natalia. Boots were made for walking. Were made for walking. Thank you very much for a beautiful presentation. I don't know if there are questions in the chat or in this room. We have a lot of people in the room now. I'd like to, well, no questions maybe. I'd like to ask you about the treatment that the Australian government is the type of treatment that the government of Australia is making with the Aboriginal people. I think the things are getting better now. Is it right?
Yeah, so there's a growing movement there to like, so they have the Australia Day there, which the day that British people got in the land. So there's a big movement to change the day because I think people are recognizing that what they did there was a horrible thing to do. So there's a book I read while I was there. The name is Dark Emu. So when British people got there, they thought Aboriginal people know nothing about the land, but actually there are some plants that they use for feeding. And when British people got there, they just ruined everything and this plant got extincted. So it's quite a story. And people are just not accepting anymore what happened to the native people there. We should do the same here in Brazil, in my opinion.
Okay. And uh, about your research, for example, you have a stupendous opportunity to go to other portions of Australia because the people that are researching there with other projects that are different from what you do.
Mm-hmm.
And can you see some similarities in between the research that you do here in Brazil, the type of research, the methodology, and the methodology that people use in the Australia, for example, or the methodology is basically the same?
This is a nice question because I would like to say that we Brazilians are doing great science here. We have amazing methods. We People see us. Well, they told me that they see us like really good researchers. So it's quite the same. I actually teach some stuff for people at the lab. So, and also something that I used to say when I was there, Diana has this, we often have week meetings at our lab, which was something that I missed a lot while I was there because it's not common for them. So it's not every lab that has this tradition of meeting every week. So sometimes I felt that I would like to talk a little bit about my project and the doubts that I was having, the problems I was having. So I think the chance that you have to work with a different group is amazing. You learn lots of different stuff. But we are doing great work here too. So people that don't have the opportunity to go overseas or to do an internship, I don't think it's they're losing something, but it's of course the experience. But in terms of methodology and results, we are really doing a great job here. I think, in my opinion, that's my vision, my view of everything.
Right? And the way they think their research, is it different from the Brazilian researchers or not? For example, a different way of thought, a different way to construct a hypothesis, or it is basically the same mechanism of thought? What do you think? Have you perceived something, anything different in this issue?
The most different thing that I noticed there was the amount of hours they work. Like they truly like to rest.
Fewer hours, huh?
7 hours a day, but they were not like workaholic there. I think sometimes we are quite workaholic here, but they work really well. The time that they are working, it's now we are working. But about thinking about their hypotheses and the research, I think it's quite the same as the experience that I have here in Brazil. I don't know if it's because Tian and Robbie has almost the same way of thinking about the questions, but I didn't find any difference regarding this. But sometimes I was there and I was focused, and then they said, "Oh, let's go have some coffee," and I was like, "No, I have to work," because I felt a pressure, because I was there with the government money, and I was concerned about everything. I'm a quite concerned person. And then they just said, no, you should rest. Let's go for a coffee. Basically this.
Okay. Thank you, Natalia. I think there are no questions.
We have a question.
Anna, go ahead.
Can I read?
Sure.
This is from Bianca Normidio. She said, have you ever tested performance of the lizards exposing them in different temperatures since the egg until they reached the adult size? Until they reached the adult size.
I tested. This is my work here in Brazil. Is this the question? Did I got it right? Because the sound's not great here, Ana Paula.
Bianca Normidio.
No, I know, but I'm not listening quite well here in my house. She's asking if I tested animals in different temperatures. The work that I'm doing here, is it?
Yes,
I got it right. So
until
yes,
they reach the adult size.
Oh no. So this is an issue. We couldn't make any of our juveniles to an adult. So we are trying to improve our animal care there to see if the animals can get to a more older age. But by this time, I think my older lizard had about 3 or 4 months. So no. But we did this with juveniles. Did I answer?
Yes.
Okay.
Okay.
Thanks for the question.
Thank you, Natalia, for your.
Sorry, we have another one.
Okay.
Just now.
Let's run the schedule.
Can I read? Sorry, the internet. Oh, okay. This is from Guilherme Hermanson. Did you test lizard body length against other environmental predictors other than the animal mean temperature?
The juveniles no, but the adults yes. And I had some results regarding precipitation rate.
Yeah, I think that is it.
Okay.
Okay.
Thank you very much.
Thank you, Natalia, for your lecture. Great job in Australia and here in Brazil.
Thanks.
And thank you also for your engagement in the protests of for the science here in Brazil. I think we have to do that kind of thing that you are doing, and I encourage the students to do the same because we are in the big crisis in the science in Brazil with so bad news these days. So you have to protest. You have to show that we are not satisfied with this kind of thing that you are working in our country. Thank you very much.
Okay, no worries.
Well, now, Tony, are you here with us?
Yes, I'm here.
Okay. Are you ready?
Yeah.
Let's go. So now the next lecture will be held by Tony de Oliveira. And Tony will present the lecture Living on the Edge, Acclimatization and Adaptation Components of Photosynthesis Temperature Dependence at the Neotropical Savanna. Tony, thank you for your presence here virtually, and the stage is yours.
Okay. Hi guys, I'm Tony. I'm PhD here. So I'll talk a little about my project and my experience that I had last year in the Netherlands when I went to Oregon University for 4 months. So I have been working in plant ecology. Actually, I have been studying functional traits using the photosynthetic traits to understand the performance of some plant species and Cerrado. So, as you know, species are exposed to different environmental pressures, environmental conditions, and to have a relationship between the environmental and the plant physiology, especially when I talk about the photosynthesis. So understand this, how the environmental influence and the plant physiology, you can understand the species persistence and productive and demographic. So and here you have a cerrado in Brazil. It's a rich neotropical savanna, and it's a large biome in Brazil. For this, you have a visible environmental gradient. Here I have two examples, the precipitation gradient and the temperature gradient. And if you look for this map, you can see that the eight or marginal areas of the Cerrado have a contrast environmental condition. For example, if you compare the Cerrado from Northeast in Brazil, you have a different annual mean temperature for the Cerrado from the Southeast areas in Brazil. For this reason, the main goal of my research is to quantify this variability of photostatic parameters and their dependence on temperature in these two marginal areas in Cerrado and Brazil, look at difference between sites, climates, variation, and the species. For this, I've been collecting two parameters, the VCmax, EJmax. They are both here to understand the net CO2 assimilation rate, and both parameters are influenced by the leaf temperature. And the first thing that you told in my project is how to measure these traits, because the most common method that the guys have been working is using the ACR curve. But if you measure different ACR curve in different temperature, you will spend a lot of time, and the problem is this process will be stressful for the leaf. So for this reason, we think about use the 1-point method. Is a great protocol that the scientists have been used for the last years to estimate the VCmax, the maximum carboxylation capacity in the field work. And they were started to test this 1 point and the different temperature. However, I need also to see the Jmax value. So you use your first ideas is to use the chlorophyll fluorescence measurements to assess the ETR, this electron transport rate. This is quite similar to Jmax value. So by look or not, in the time when I was talking, I think about this new protocol, the Professor Elma Vandevoort, sent me an email from me and the Professor Thomas, talk about the possibility, the internship in the Vargas University research. So she sent us this call, the Vargas University Graduate School Sandwich PhD program. It's a program for the Vargas University that has a goal to invite students, PhD students from different countries in the world to take some time in Vargas University and develop part of their PhD project there. And the Vargas University is one of the best universities in the world, actually. And for example, this is the first agricultural university, the number one, and the agricultural university, and the second in the environment and ecology. And you have a lot of scientists that have been working with photosynthesis. And for this reason, we started to look for some collaboration there to help me to improve my skills in chlorophyll fluorescence. That's the new stuff for me. So to start my PhD sandwich there, I need to apply for this call, and it's a easy this process. I think you need only a letter with a collaboration between the professor from Vargas University and the professor, your professor in the home institution. It's easy because Professor Tomás, my supervisor here, has a years of collaboration with Professor Elma Vandevoort from Vargas University. It's for me. You need later motivation, your CV, and your project, of course. If you contemplate with this program, you have a grant when you can stay there for 18 months with a mensual scholarship, and the all travel cost, including their visa application, is covered by this grant. It's an excellent opportunity. This is the Vargen University, part of the campus, actually. Vargen is a small city in the middle of the Netherlands, around 40 minutes from Amsterdam. It's like a university city. You have a lot of students there. It's an international university, so you found students from China, Africa, and South America. Anyway, this is the Lumen. Lumen is a building when I was working for a month there. During my time in Netherlands, I was working with the Plant Ecology and Nature Conservation Group. It's an interdisciplinary group. It's amazing it's amazing people. They have helped me a lot with my English and with my research. I have people from different countries, as I told before, and in this department you have a Brazilian guy from Viçosa. You talk a lot about your life and Brazilian problems in the beginning. This is the Professor Elmer. Elmer is he's an ecologist and have experience in savanna areas. They have been working with savanna and transition areas for many years, and he is my main supervisor there. And this is Professor Jeremy Herbison. I met Professor Jeremy in Netherlands, in Vargen. Professor Jeremy is an ecophysiologist and have experience in chlorophyll fluorescence measurement, especially looking for health is warm stress and the chlorophyll fluorescence process. One thing that made my eyes shine is the huge greenhouse complex that you have there. It's amazing. You have a crops, of course, because the agricultural university, but here you have a lot of different kind of gardens with species in different parts of the world. In my case, for example, I started to measure my chlorophyll fluorescence in the tropical gardens. And when I tested my 1-point method in my VCMAX, I stayed there for 1 month with Professor Jeremy and Professor Bart. Professor Bart is another scientist that has collaboration with Professor Tomás Domingues here. And in this period, I learned a lot about the chlorophyll stuffs. And when I did some adjustment in my protocol that I have been using in my field work here. As a result, you have a good relationship with my new protocol, with the common protocol. I compare here the VCMAX, MIDIMAX from the ACI curve with the VCMAX ATR JMAX actually from the 1-point method, including the chlorophyll fluorescence. I have a great relationship. It's a good thing for my work there. During my time there, I took some course, especially the statistical course and the model design course, because the Varninges has a real they like to be sure if their students or their research will be a great field work. So the most part of the course has focus on the experimental design, but you can take any course there. For example, you have a course for how to talk and how to talk in public, or how to write a good papers. You have a lot of type of the course. It's a great thing that I found there. Another thing that I really like in Vargen University is because every week you had a PhD meeting when you can share with the professors, a postdoc and PhD students your project, or some data analysis or some graphs to interpretation. It's an amazing opportunity because you have people from different areas. And for example, for my project, I receive a lot of good collaboration because you have a geneticist, you have people that have been working with modeling. It's a great time, and I learned a lot during this meeting because you have a different staffs in this department. For example, I was the only guy that working with photosynthesis, for example, but the most part of the guys is like the geneticist. Another thing that I think is important to show you is because they are very careful with the PhD mental health. And during the day, you have to stop your job, your work for 3 times on the day, because you have the coffee time, the lunch time, the tea time, when you talk about your life. You can't speak about your project. You need to talk about your life, your problem, how are you doing, how are your family, and talk about the Brazilian football. Actually, when I went there, everybody wants invite me to play football, but I don't like it actually. Uh, as I told before, you have students for different areas, different countries, and in the Varning. So I think the most part of the student feel alone there. So you had a group of students for different areas: the Germany guys, the Portugal, and the French. So every week or every 2 weeks, you meet together to cook it together. You have international dinners or Brazilian dinners or Chinese dinners. When you cook it together, a drink and dance, it's actually it's amazing time because you can relax, you can because I think most of these people feel that studying learning you away of your home country, for example, it's a big pressure. So they have this way to be this process easier. So this is Vargas University. I stayed for only for a month, but I want to come back soon there. So, but you have a lot of Brazilian guys there. You have programs, you have a lot of collaboration between Vargas University and Brazil. You have the USP, Unicamp, the people from Viçosa and Brasilia. So actually, I think the Vargas is a Brazilian home and the Europea. I think I felt me home there. So I think it's a great opportunity if you want to apply for this call, and you learn a lot about the new cultures in different countries in the same place, and you look for a different way to make science. One that the focus is not only the papers and not only the result, but the paper result and the good scientist and the human scientist. It is. I would say thank you for Thomas for the opportunity to make me enjoy this journal, and I would say thank you for everybody from the program and the Vera and my friends here. It is.
Thank you, Tony, for your lecture. Are there questions in the chat or here in the room? No questions in the chat. Guys, anybody? Because we have a lot of people here. And it's good. No? Tony, I have a question of my own.
All right.
All right. Thank you for presenting this university. It's an amazing place to work
Yeah.
in with this, Natalia. And the buildings, all the structure, the architecture of the buildings, all the buildings are very clear. That greenhouse was complex. It's amazing to see that.
Yeah.
It's almost like
I
a
love it.
dream. It's almost like
Yeah,
a
yeah.
dream. Uh-huh. And I'd like to know how can you handle your material? Because you have some materials that have to be sent to Waningen to be analyzed, or the
Mhm.
analysis they were carried out here in Brazil, and you also get the numbers or do the maths in Netherlands.
In Netherlands, actually, I did only the tests of the methodology because I want, as I say, I want to improve my skill in the chlorophyll, for instance. But my tests and my data collect, I did only in Brazil. So the materials I use only this one leaf, for example, by individual. I actually don't use any data from Wageningen University. But if I come back, probably I will. My Jeremy asked me to leave some samples. I don't know if it will be possible to try the plasticity and this response, because you can grow this species in different temperature in different greenhouse there. But there I work only with some local one or two individuals, one leaves, just to try to learn about the chlorophyll measurements.
Okay. And about the people from Netherlands, and you say that you are very focused in the work, but there are small times in the day, 3 times in the day, that is forbidden to talk about your project, your job, and you have to concentrate in the personal facts of your life. Yeah.
Yeah, on Friday, every Friday, I think you play some game. You bought some beers in the side of the build, and you always did a happy hour before the work. It's amazing. It's amazing experience actually, because I believe in Brazil you have a fox, and once you need to produce, you need to collect data, you need to study, and they're not the people have the focus, of course, but you want to enjoy the company of the colleagues. It's amazing.
Okay. Is the beer good?
Yeah.
Okay. I think there are no questions. Okay, Tony, thank you very much for your lecture. Congratulations for your work, your internship in Wageningen, and I think Thomas has a very good student in their lab,
Oh,
in his lab.
thank you.
Thank you very much. Well, after Tony's lecture, there is another lecture left from by Cynthia Neves de Souza. Cynthia, are you there, or are you here?
Hi, Professor Milton. How are you?
Is everything okay to your lecture?
Yes, Professor. But I have problems my internet connection, so I need to cancel my camera interference.
Okay.
Okay,
Do
okay,
you
no
want
problem.
you want to go to outside
Interference.
the room and after to return?
Yes, I think that's better. Close my camera and just audio now.
Okay,
Okay?
as you wish.
Thank you. Thank you.
So the next
I will check.
lecture, okay, the next lecture will be by Cynthia Neves de Souza, and the theme is botany, reference herbarium of Gesnerioideae, Gesneriaceae, visit and collect of pollen and floral material in Switzerland and France. Thank you, Cynthia. The stage is yours.
You're welcome, Professor. I will share my screen now. Can you see? Can you listen to me? Good.
Professor? We are listening, but the screen doesn't work.
Okay, I will close my camera. Sorry. And now my presentation here. It's okay?
No.
No. Oh, sorry. Apresentar agora. And now?
Working
Yes.
finally.
It's going.
Yes?
Yeah.
Yes, good. Thank you. Thank you. Here. So, hi, everybody. I hope you are good. My name is Cynthia. I'm PhD student in the program in Comparative Biology. I'm a botanist. And my research is conducted in Lampali, Laboratory of Plant Morphology and Palynology, in Jabuticabal at UNESCO. So, put laser here. My Brazilian supervisor is Dr. Eduardo Custódio Gasparino, and my supervisor abroad, Dr. Alan Chotin. And the title of my presentation is Reference Herbário of Gesnerioides gesneriaceae, visited and collected of pollen and floral material in Switzerland and France. Later, I will give more details about the supervision given by Dr. Alain, the main supervisor of this project abroad. My group of study is Gesneriaceae, has about 150 genera and 3,500 species distributed in tropical regions with a few representatives in temperate regions. The main centers of diversity are located in northwestern parts of South America, for example, here, Colombia to Ecuador, and also southeastern parts of Brazil here. And this family is divided into 3 subfamilies. Here we are Sanangoide, Gesnerioide, and Didimocarpioide. And my research is focused on Gesnerioide here, this group. There is an image of conviolet, one of the ornamental species of Gesneriaceae. This is very common in Brazilian supermarkets, for example. And the Gesnerioides subfamily contains 75 genera in more than 1,200 species, distributed exclusively in the Neotropicus, this image here, except Titanotrichus genus, with a single species found in China, Taiwan, and Japan, not represented here in this clade, and some Pacific taxa, such as Mitralia, here in red. This clade has wide distribution in Neotropical region and high morphological diversity too. Here, it's possible to observe the diversity of Gesneriaceae in general, but also in Gesnerioides subfamily, as diversity of shape, colors, and detail in its flowers. Now, my field of study is botany. In botany is palynology. Palynology is the science that studies the external morphological characteristic of pollen grains and spores. The pollen grains are characterized mainly by the type and the number of apiary and the exine ornamentation, and they become tool of taxonomic value that help us to identify or classify the species. For example, in this scanning electron microscope image, it's possible to see different characteristics of pollen grains, of course. For their size, here it's possible on the same scale, we can see difference in size between them and many other characteristics. So my research is about the analysis and description of polymorphology of gesneriid species to discover the morphological diversity of pollen grains of this large group of neotropical species and to assist the taxonomy of this group. Choosing to study a subfamily in 4 years of doctorate, we need to think about how many species will be needed. So how many species, where, and how to collect them. So there are 2 ways to collect species for palynological analysis. The first is field sample, and the second herbarium samples. For example, here. However, field work is not possible when studying a large number of species. The best option is, as here, desiccated material. The definition, the herbarium, is a collection of dry and pressed plants that preserve snow and new species of plants and fungus too. Herbarium is useful for us to obtain official information about description, classification, and organization of these species. The great taxonomist of Gesneriaceae species, especially of the neotropical species, is Dr. Alain Chotin, associated researcher at Conservatoire Jardin Botanique de la Ville de Genève. Alain has been working for more than 40 years with Gesneriaceae. He has made numerous expeditions to Brazil. He's passionate about our country. And our contact started because Dr. Alain Chotin was co-supervisor of Dr. Eduardo Gasparino in his thesis. They have been in contact for many years. Here, I brought this example, Nematanthus. It's a genus of study for Alan's thesis, and a great information now, if you want to know more about the floral diversity of Gesneriaceae, I invite you to visit the Brazil Plants website here. It's a fantastic collection created by Mauro Peixoto. He's an excellent cultivator of Gesneriaceae. This is an important website, and there is part of in Portuguese and English too. Alan, I said association, he's associated researcher at Herbarium G. This Jardin Botanique has about 6 million specimens. The Herbarium G rank among the most important botanical collections in the world. It includes plant and fungal specimens, and a special emphasis on the Mediterranean area, near and the Middle East, South America, and Europe. Some photos there, a beautiful place. So in 2019, I had my collection project approved by São Paulo Research Foundation for 3 months. It was very difficult, but after much patience and perseverance, FAPESP finally approved my project. My period there from June 1st to September 1st, 2019. About 120 samples of species were collected. So about 200 samples of specimens there were collected. And Alan was always by my side, my nice friend. And the Botanical Garden has an important scientific and public collection of Gesneriaceae. I brought some images for you. I collect samples from cultivation and also material fixed and 70% alcohol from many expeditions carried out by Dr. Alain Chotin and Dr. Mathieu Perret. There is a beautiful species, Coleria amabilis, cultivated there. I visited two other institutions. One example is University of Zurich. Herbário Z is a union between University of Zurich and the Federal Institute of Technology in Zurich too. And Dr. Anthony Feller hosted Alain and I there for a couple of days. And then samples were collected in Herbário Z, and 7 samples were collected in Cultive. This is image, it's great because the greenhouses looks like UFOs. And I brought 2 examples, Sininja gigantifolia, my hands now, the big leaf, and the beautiful Sininja ovulata. Herbário P has a large collection, around 6 million specimens. It's a general herbarium. And Peter Philipson is a nice American. He hosted me there with enthusiasm, even with my limited English. And so nice. In the museum, in the Herbário P, has about 10,000 specimens, but I collect only 28 samples because there are rules for sample collection. Firstly, only a limited number of samples can be collected, and secondly, I'm not allowed to collect the samples. I just select these cases, and I staff from the herbarium collect the samples for me. And then he sent to Alain in this case, and Alain sent to me here in Brazil in November 2019. Let's go. When I received the invitation to be a speaker at this event, I thought a lot about how to share my experience abroad, because personal experience, that real experience, for example, leaving our country for the first time, listening to many different languages, feeling unsure when speaking another language. It's really hard. And so I thought I need to share with you, because sharing, in my opinion, It's an important point. It's good to know we are not alone and that we have the same experience, I suppose. So I will share with you my experience when I lived in Brazil for the first time. I went to Switzerland with very limited English, and I still have limited English, but their explosion, boom, the four official languages and the English is not among them. The Swiss people speak mainly French, Italian, German, and Geneva is a very diverse city in terms of cultures. I remember the first time on supermarket, I heard about 4 languages. My ears were hypersensitive. But in Geneva, people speak mainly French. Then my experience in my hand became a total mess. I was tired with a lot of information in my head, but it wasn't an easy experience. But I have many stories, mainly the typical experience of meeting Brazilian people, everyone, for example. But it's perfect experience for me. In the last days in Geneva, Professor Fred Stauffer at Herbarium G asked me, Cynthia, how do you feel now after the experience here? And I remember that I said that my field of vision has changed. Now I see a world with short distances, very close. Everything is possible. So we can do anything we want, just believe it. Shared experience can scare us, and for us to turn 30, same se virar nos 30, but in the end, I'm sure your field of vision will be bigger and your perspective on life lighter too. I believe that. Sorry, motivation talk now, guys. Finally, I can visit many places, like the house where the Condor's generation lived. They were Swiss botanists from Geneva, their house here. And I saw the house where the Einstein lived for a period in Bern, in Switzerland. And Alan and I did many tours. He's a great guide. And I also fulfilled my dream of visiting Paris, Eiffel Tower, Café by Amélie Poulain, from the French film Le Fabuleux Destin d'Amélie Poulain. It's great. Louvre Museum. So great opportunities there. And again and again, I learned I shared a great food and drinks. Of course, craft beer, I love it. And Alan is a master chef. I was very lucky there. And so guys, finally, this experience was only possible because people believed in me besides myself. Therefore, I would like to thank you so much, Dr. Alain Chotin, my friend, immensely for making it possible. And today I can be here, share my story with you. Professor Eduardo Gasparino, thank you so much too for believing in me and my work over these 10 years together at Lampal and personal life. And the Professor Fred Stauffer for making my collection in Paris possible too, and share a delicious Arabica lunch in Geneva. And thank you so much, São Paulo Research Foundation for funding, and the Herbarium GZMP for allowing my collection. So thank you so much for your attention, and I wish you the best, always. Thank you.
Thank you, Cynthia, for your lecture.
Thank you.
Finally, I've seen flowers this day.
Yes.
Orchids yesterday, and now Gisneriaceae. Are you going to turn on your camera, or?
Okay, sorry. Yes, I will try, okay?
Okay.
Hi, guys.
Okay, thank you, Cynthia. I don't know that. I think your YouTube broadcast is not working. Is it correct?
Sorry?
Yes, it's not working.
Oh, sorry. Okay. I don't know if there are a few questions in people in the room. Anybody?
The final presentation.
Yeah.
Milton, can I ask a question? Speaking, not writing.
For sure. Go ahead.
Cynthia, I believe you heard from lots of people because you're Brazilian. I don't know. I had this experience, so they look at me and say, "Oh, amazing fauna and flora there in Brazil." So you went to an herbaria. And I would like to know if people have this view of Brazil also there, if they were asking you about the Amazon forest and stuff like this.
Yes, of course. The good question. It's very common, this question for us, Brazilian. And I had experience in Herbarium P, because Peter asked me, Cynthia, you went to Paris here to collect some Brazilian species. So you went. Sorry, my English is bad now, but the type. Eu saí do Brasil e estou agora em Paris coletando espécies brasileiras. It's an example about my experience. I don't know if... yes, thank you. Thank you for your question.
Cynthia, I would like to know if the curators... Yeah, I'd like to know if the curators are happy conceding you the material that you are studying.
Sorry, Professor?
Sorry, slower. I'd like to know if the keepers, the curators of the collections, if they are happy that you are taking material from the specimens to be studied.
Yes, of course. I love it.
Okay, but they don't like when you pick up some flowers.
No, Professor, no. I hate me.
No, I'm a keeper, and I know that sometimes you have to give flowers and fruits to other visitors, but we don't like to do that.
Yes, I agree.
Okay. Thank you, Cynthia, for your lecture.
You're welcome, Professor.
Well, Geneva is amazing. Yeah, Geneva is amazing. I don't know Switzerland yet. I know France, but not Switzerland. and it must be a wonderful place to visit too.
Just to go there.
Yeah.
It's a beautiful place.
Okay. Thank you very much, Cynthia.
You're welcome.
Well, Cynthia's lecture was the last lecture that I have. We are with some problems in the YouTube broadcast, and if you could share the link of the room with your colleagues, it will be amazing because we have 36 people here in this room, and I know that there are more people than it in the YouTube. So I think you can open the link also to your colleagues that are not from the program, because we have faced some problems in the YouTube broadcast. Okay. Well, so after the lectures, we are going to the final part of our meeting of symposium, and I'd like to invite two people, the students representatives, Ariadne Dalacqua Aires and also Ana Paula Cipriano, to talk for a while with us, because I think it's important, very important, and I have stressed at this point that the students could be listen also in this symposium, and I invited Ariadne and Ana Paula to talk a little bit. Thank you, Ariadne, Ana.
Thank you, Milton. Good afternoon. It was a pleasure to be on this event and an opportunity as a training research to hear the experience of different biology research. This type of scientific events, I believe, provide the program are important to expand our knowledge frameworks of the three different areas that exist here, and speaking not only of the symposium, we also think that the quality of the program allow us to have many opportunities in addition to an excellent academic formation right here. So thank you very much.
Yeah, good afternoon. And we also believe that the broad vision that we obtain within the program, learning not only for our project, but also for other research of other areas, provides a lot of experience to work in the most diverse biology areas. So, in addition to learning a lot from the symposium lectures and videos, we are also studying an extremely qualified program, and even now, in these difficult times for our society, we are working so hard to keep our science alive and in constant construction. So I just want to thank you all for the participation and to be here and asking and, giving this good experience for us all. So I think that is it. I just want to thank Milton and Daniel, André, Ludmila, Vera.
Okay, Ana Paula, Ariadne, thank you very much. As I think everybody knows, Ariadne and Ana Paula, they are the student representatives of the course, and I would like to stress the fact that it's very important to our students to be represented in our committees. And every time we have a chance to take part in these committees, it can be in the CCP, in CPG, or in other committee, it is very important to the students to be listened in all aspects of the programs of the university, and also in aspects of the research that we conduct here in our university. So the participation of the students is crucial to the development of the program, development of the university, and of course, the development of the society as a whole. So I would like to thank Ariadne and Ana Paula, because they have a lot of work doing this kind of thing, be representatives, because we have to listen one part and you have to talk to the other portion of of the system, and it's a kind of link that's very important to be made. Okay, so encourage other students to take part in the to be representative of the students anytime it is required. Okay, thank you, Ariadne. Thank you so much.
Yeah, I'd just like to say that it's open this registration for the new representative for the students. So please sign it.
Okay. Yes, we have to stress this fact. Thank you very much. It's quite important to the students and our program, the representatives of the students. Okay. Now we are going to the final, and the thing
Mute.
that you are sorry.
Oi? May I say some words?
Well, if you want to speak in Portuguese.
No, I would like to speak in English, my poor English.
Okay, let's
I,
go.
I just want to say, even though I'm not a student, but I'm so pleasure to make part of this program and be able to collaborate with all. I don't know if you understand my poor English.
Oh, Vera, your English is perfect, and we have.
There is a long time that I study. I have not studied, but I love, and I love everybody that I do. I love my all students. I love you too. As my aos recadinhos. Ai, gente, don't be me desconcentrated. No, but I'd like to say thank you for you to understand, to help us. Even though this time, everybody is so tired, so confused, but I guess if we can help each other, you can go already. I guess it's exactly what that's what I showing everybody together we can do everything.
Well, thank you very, thank you very much for words. I think all the program have no words to thank you because all your work, your attention, your desire to help all the time, every time, never tired. So we have just to thank you all your passion on the program because you are a passionate person, and has been a pleasure to work with you. And I think it is has been a pleasure to all the students also be to have you as our secretary helping them every time. So I have to thank you very much.
Thank you.
Everybody love everybody loves Vera. Thank you, Vera. Well, now we are going to our video award section finally, and then the closing ceremony. I have some few words to say in this video award section. I would like, I think almost everyone every time, but it's necessary because when we thought about the video section and the videos that you have to make to our channel, I know that it will be challenging to do that kind of thing, but I was sure that the products that will be held for by our students will be good, and in fact they are excellent videos with excellent way to talk with the society, to talk with a broader audience. So I'm very happy and very proud of you because all the videos are really amazing. Okay, thank you very much. And I was concerned because maybe the students will not bought the idea to go to make the videos, but the videos are amazing. And I hope that you have the chance to develop other skills than those that you have already. So thank you very much for your efforts to make these videos. They are excellent. So we are going to the videos award. Think about this as just a joke, a celebration, just to get a little bit of emotion in this ceremony. And I will present my screen, share my screen a little bit. Let me see. Are you seeing my screen?
Yes, we are.
Paloma's baby, yet. So cute. Let's go. So this is the third symposium in comparative video awards. And this is a kind of celebration. That's a kind of joke, just to have some fun in the final of the symposium. This is the third edition of this comparative biology awards as well. And I will explain a little bit the mechanism, the methodology that we use it, just to remember, to remind to you. We have 46 videos that were rated, 44 that the videos are public, and 2 are private because the authors asked me to do that. And they were divided by author's statement in the 3 main lines of research of our program. This division was the the authors did their opinion about the line of research that the project belongs. The 3 main lanes of research are evolution of biodiversity, ecology and conservation, and history, philosophy and teaching of science and evolution, right? And another methodology that we used, each video were rated with at least 5 evaluators, okay? 5 people evaluate each video, okay? Well, some videos were evaluated for more people, but at least 5 evaluators to each video. We divided the awards, and so we have 20 awards, and we divided the prizes because we have a kind of asymmetry in the projects that we conduct in our program, because we have much more projects that are focused in evolution of biodiversity than in ecology and conservation and in history, philosophy, and teaching. That's the reason that we divided rating the videos, and we conceded more prizes to evolution of biodiversity than to ecology, comparative ecology, and history, philosophy, and teaching of science. Okay, so we trying to balance the number of awards with the videos that we received and the prizes, right? The prizes. The prizes are 100 reais in vouchers on Amazon and Amazon.com. So we will gift 100 reais in vouchers, and the nominated and the winners of the prize can exchange this 1 reais in products of Amazon. And this prize is equal to everybody, no matter if the video was prized as a first one or in the 5th place. No matter, this prize is equal to everybody. Okay. Well, I divided also the winners for the three main research lines of research of the program. So first, the announce of the winners in the history, philosophy, and teaching of science. In this line of research, two students were awarded. But first of all, I would like also, I forgot to say that I would like to thank you again for the effort to do the videos. Some of the videos, they were produced in another language, for example, English. So we have the Jessica Gallo, Alexander Brunello, Tony Oliveira, Guilherme Hermanson, Silvio Nares, that uh, made the videos in English, and one video in Arabic, Wafa, that is Syrian. So I asked her to do the videos in Arabic. Well, finally, the winners in this is in second place, Gabriela Cristina Scanzella Iglesias. This is the number of the video. Let me see. This is the number of the video, and this is the rate of the video according to our evaluators. Okay, so the second place, Gabriela Cristina Iglesias, and Fernanda Gonzalez Arcanjo, winner the first the first place. So congratulations to Fernanda and also to Gabriela for the videos and for the awards. But all the videos were amazing as well. The rates of the videos were very, very high in all the categories, okay? And some of them very close, the concepts. The winners in the ecology and conservation, also in this line of research, there are two winners. Tony Oliveira in second, and Eduardo Haberman in the first place. Congratulations, Tony and Eduardo, for the awards in the Ecology and Conservation Video Awards on our Comparative Biology Symposium. And finally, the winners in Evolution of Biodiversity. In this line of research, we have 8 videos that have been awarded. First, the 4th to 8th, Malu Araújo Almeida, Silvio Onari, Stephanie Liao-Kang Goicoechea, Ana Paula Cipriano, congratulations, Ana Paula, and Gabriela Baroni Leite. As a highlight in this page, I would like to highlight the works of Malu, that was amazing, with a kind of cartoon. Silvio Nari, Silvio is really a YouTuber. He has a talent to be a YouTuber. Stephanie is, that's incredible, the social, the engagement in social media that Stephanie has. It's amazing to see. And also Ana Paula and Gabriela, because they have a very good presentation of the ideas, the thoughts, very organized videos. They are really amazing. And the first, second, and third place are mother, Paloma Gaspar Torati. Natalia, congratulations, Natalia, for your second place. And in the first place, Felipe Pisaneski. So you can see that the rates were very close to each other. And I would like to congratulate all of you for the effort to do the videos, to making things, the audio, the subtitles, all the things that you have to perform and to learn to do these videos. I don't know if who is here in the class. Malu is here. Congrats, Malu.
Thank you. I'm happy. I'm very
Silvio.
happy because I had a lot of work.
Silvia is here also. Natalia. Where's Felipe? Felipe is here also.
Yes, I'm here, Milton.
Congrats, Felipe.
Thank you.
And I will not say that Felipe was asking me about the video one day. One day to go the period to send the video. Okay, Felipe?
Yes, yes. I had some problems.
Okay, guys, thank you very much. It was really good. You produced very good videos. All of you produced very good videos. I know that you said bad words about me. Okay, no problems. But I think the final product that you made, our final product is amazing, and you can, for example, show it to your grandmother, to your father, to your mother. That's the thing that I do in the university. So I am able now to explain you what I am doing in the university. So thank you very much for you. And that's it. So we have to arrange the things to conceive the prize for you, okay? But we are keeping touch to the vouchers in the Amazon.com, okay? And this event is not sponsored by Amazon.com, to be clear, okay? Unfortunately. Thank you, guys.
It would be interesting if it wasn't sponsored by Amazon.
It would be amazing, Silvio, if you have a contact with CEO of Amazon or executive in Amazon, please tell me. That will be very good.
I will do that.
Okay.
For the next symposium.
Thank you very much. Okay, guys, this is just a joke, just to have some fun. Okay. I was very concerned, as I say, that you will bought my idea, the idea of the coordination, but you bought the idea, and I am very happy with that. Thank you very much. Now we are going to the closing final words. I have some words to say to speak. First of all, I think we had 2 days of much work to me, to our organizing committee, and again, I would like so much to thank our organizing committee, Professor Tomás Domingues, Danilo Trabuco Amaral, that take care of all the transmissions and all the things in the broadcast of this event. Andrés Guicero, that helped a lot and produced the book of abstracts. So this is the first time we have a book of abstracts in the symposium. You can see the abstracts that you made, that you written, that you wrote, put in the LATS curriculum LATS. So it would be amazing to you to see this kind of thing. Ludmila Micheleonis Pesarin, also by her help. The student representatives, Ariadne, Ana Paula, and of course our very special secretary, Vera, Cassia Cicilline de Luca, always kindly, always lovely, and always wanting to help everybody. Okay, thank you very much. Of course, I'd like to thank you for your patience, for the things that we saw in this event. I'd like to thank the lecturers, the students that lectured, the postdocs that lectured as well, and our invited speakers, Professor Carlotti, Paul Fine, Professor Charbel in the first day, and Paul Fine. Today, Professor Jonathan Cott, and also Professor John McNamara and Professor Christie, that be present in the opening ceremony as well. And the final words, I think we have products that are very good, the book of abstracts and also the videos. And I would like to ask you to share the videos to your colleagues, to share the videos to the people that you know, to spread the things that you produced, okay? In the beginning of in the opening of the ceremony, I told you, and I said that the symposium is a kind of a meeting, of course, and it's there were some objectives in the symposium. The first one is to be a facilitator. I'll read that. The facilitator of international exchange, helping to improve communication in English, since all the activities are conducted in this language. And also, this symposium is an opportunity to show to ourselves, to everyone else, to students, advisors, what kind of research is conducted in the program. And that this event provides a meeting point for our students who can thus exchange experience in their research and interact with their colleagues. But there are two other things that I would stress now. One of the things that when we thought about this symposium these days with this pandemic and social distance, we were and we are very concerned with the health, mental health of the students, and some feeling of loneliness of the students. So we thought this symposium as an opportunity to bold the students. And if you think about the video, the activity of the video producing, that course that you made to produce the videos, these were opportunities to bold the students, to put the students working together, to talk to each other, to a little bit overcome, to cope with the loneliness that we are facing. So it was an opportunity also to put the students together in this period. Okay, this is the first thing that I would like to say. And now I think there was another objective to this symposium to happen, that is to celebrate. I think we have to celebrate one thing that is very important to us, that to celebrate life. We have to celebrate that you are alive, because many people in these days are passing out, many people are sick, and others are unable to attend a symposium or a conference or a meeting, I don't know. And we are alive here, and we are doing our job, doing our things, and I hope in the near future, all the things that we consider as normal things in this life, like, for example, walking the street, going to a party, drinking in a party, or only meeting their friends. I hope for the deep of my heart in the near future, we will be able to do that things, and the normal life will resume as soon as possible. That's the desire of the deep of my heart. So if you have to thank, if you have to thank your God, thank your deity, thank your relatives, your partner, thank science, do it, because we have to think that we are alive in these days. So thank you very much. And now I would like to ask your permission to talk a little bit in Portuguese. So I will switch to Portuguese. Foi prazer ter vocês aqui no simpósio. Fiquei muito feliz com a presença virtual de vocês, ver vocês de novo, foi muito bom. Eu espero que todos estejam também preparados para ir no 10º encontro de Biologia Comparada, que dessa vez vai ser em virtual ainda, mas eu espero que daqui a pouco todo mundo vai estar com a vida normal, fazendo as coisas que gosta, e a gente vai sair sem máscara, e a gente vai ser feliz, e tudo isso vai acabar. Tá bom? Obrigado, gente, foi espetacular, agradeço demais. E eu queria fazer um último pedido, para a gente abrir as câmeras para a última fotinha, se vocês puderem.